Assalamualaikum and hi to all examiners. I'm Farah Alia binti Hassan with matrix number F18B0029, a final year student of Bachelor degree in Animal Husbandry. Today, I would like to present my final year project proposal on antibiogram of Edwardsiella tarda, isolate from disease ornamental fish of aquarium shop. Before we go through on my final year project proposal, this presentation will cover on research background, problem statement, objective, literature review, methodology, expected outcomes, and lastly, timeline of this project. When we look our eyes on ornamental fish, the first that caught our eyes is their uniqueness in the colors and the shape that makes an aesthetically pleasing to make a fish keeping or hobby. Therefore, the ornamental fish breeder will produce mass quantity and better quality ornamental fish for the aquarists, where there will be an export and import activities, which is important for our economic values. Many aquarium shops in Malaysia are selling freshwater ornamental fish because of its easy to be bred, low cost in farming with beneficial of our country that have tropical climate that can produce high quality, colorful fishes and high value. Yet, bacteria disease can cause great losses and harmful to these ornamental fish. Many studies shows that these bacteria mostly found in freshwater fish that can cause infections and mortality with severe liver cirrhosis to human when consume this fish. Yet, there is a journal shows that Edwardsiella tarda with other bacteria occur in freshwater ornamental fish that purchased at the aquarium shop. Therefore, this experiment is meant to find the validity and further analysis and investigation on identifying Edwardsiella tarda on disease ornamental fish or from aquarium shop. This experiment need to give high attention and prioritize because today ornamental fish are highly demand in market and can cause great losses to with intensive culture farming with infected bacteria of Edwardsiella tarda. Because of ornamental fish are more sensitive with artificial environment, by trying to find the cause of the great loss towards the ornamental fish, we can prevent ornamental fish culture from infected and achieve optimum survival rate. The purpose of this experiment is to isolate and identify Edwardsiella tarda from ornamental fish and to characterize antibiogram of isolated Edwardsiella tarda. Next, the significant study of this experiment are Edwardsiella tarda is a harmful bacterium and can have potential cause in human health and other aquatic animal. And it is crucial in raise awareness on Edwardsiella tarda in ornamental fish. These are few reference. From here you can see these are few reference, article and journal for my literature review to ensure that my project are tally and information is not overlap with my finding and during conducting this experiment. In this experiment, the methodology are sample collection from diseased ornamental fish will be made by collect the sample from liver, kidney, and external lesion of the diseased ornamental fishes and culture on alkaline peptone water with 1% of sodium chloride with pH 8.6. Then isolate the bacterium into xylolysine deoxychocolate for 24 hours at 35 degrees of incubation. Next, bacteria will be subcultured in tryptic soy agar for another 24 hours at the room temperature. Bacteria identification using BBL crystal kit will be done by place whole panel inside incubation at 24 hours for 35 degrees to 37 degrees Celsius. Then, the test of idle and oxidized will be performed separately using idle reagent and BBL oxidized reagent. Record data will be done by see on the color changes in BBL crystal after 24 hours by place down. Next, antibiotic sensitivity test will be done to evaluate resistance of antibiotic, followed by calculation of multiple antibiotic resistant mRNA index. Towards at the end of my experiment, I am looking forward on isolation of Edwardsiella tarda is present in diseased ornamental fish, especially in freshwater ornamental fish, and looking forward that this experiment will be helpful to other researchers improve on their fish health. Lastly, here are the timeline for the whole experiment, including producing proposal, making the experiment, result, and complete the thesis. Despite Malaysia control order, the duration timeline needed will be from March 2021 until January 2020. From here, you can see these are few reference, article and journal for my literature review to ensure that my project are tally and information is not overlapped with my finding and during conducting this experiment.